So thanks, Francis, for the invite and opportunity to present in this seminar series, and thanks to all of you for showing up. If anybody knows me, they know that when I have to do a presentation, I agonize over the slides, and I get super nervous for days and days beforehand, and then usually what happens is I go and I give my presentation, and it's like an audience of 5 people. So thanks so much for showing up. You guys all made my agonizing totally worthwhile. Okay. So today I'm going to talk about some of the above and below ground connections, important understanding forest die-off with some implications for seedling establishment. So globally, forests cover about 30% of the land surface area. And so forests, they provide a variety of ecosystem goods and services. So they're really important for carbon storage. They're really important for habitat for all the different species on the planet. But worldwide, what we're seeing is this phenomena called forest die-off. And so on every continent, there's evidence that forests are receding. And so a lot of these reasons have to do with something to do with the climate. So for instance, in North America, let's see. Oh, there we go. Okay. So for instance, in North America, in southwest U.S., Brashears et al., they documented that about 1 million hectares of pinyon pine were lost during a drought that lasted a few years early in the decade. So closer to home in the aspen parkland, we've seen widespread dieback or die-off of aspen. And so this has happened across Alberta and Saskatchewan. And this happened in sort of in that same drought time, 2001, 2002. And then, of course, so there's these climate-induced reasons for forest die-off, so things like drought. But then, as Francis said, there's also things like insects, right, that are also related to the climate. And so we're all familiar with the mountain pine beetle epidemic. So we're looking at here is a map from the 2010 Red Tree Survey. So each year the provincial government goes out, they fly over the province, and they ID where they see red tree attack. And the red trees, they just indicate, so this is a place where mountain pine beetle has come through. And if you include in Alberta, over 14 million hectares of lodgepole pine forest has been killed by mountain pine beetle in the past decade. So I know most of you in this audience is going to be familiar about sort of the biology, the interaction between mountain pine beetle, lodgepole pine, but I'm just going to go over this very quickly. So what happens is you have a beetle, right, and it attacks a green tree, and she bores into the bark. It's a bark beetle. This happens in the summertime, and then they emit this aggregate pheromone that basically invites a whole bunch of beetles to come mass attack this tree. The beetles mass attack the tree. This exhausts the tree's defense. The beetles, they also carry several species of fungi in their cheeks, and so they go into the tree. This fungi is released. It colonizes the xylem, basically shuts down the water flow. As the eggs, they turn into larvae over the winter. This girdles the tree, and sort of through all these pathways, the tree finally dies. What we see, if you're looking from the outside, is that, so you start with this green tree, the beetle comes in, and basically this tree just really, really dries out. So the needles all turn red, moves into this red attack phase. Over time, the needles fall off the tree, and then we move into this gray attack phase, and then finally more into this advanced decay. So this is the time where like twigs, branches, limbs, and all this stuff are coming off the trees. But what I'd like to point out is that so this disturbance caused by mountain pine beetle, so this is different than other disturbances we see in the boreal forest. So what happens, so we have the beetle come through, kills a bunch of trees, but those trees, so they remain standing on the landscape for quite a long time, right? So they're killed really quickly, but they stay on the landscape for a while. When you look at the understory, basically that understory also stays intact, so nothing really happens to it. And this is really different than other common disturbances we see in the boreal forest. So for example, logging and fire. A fire will go through, and there often will be stems intact following that fire, but the fire really burns out the understory and the soils in that site. And it's also different than logging, obviously, because logging just will take off all that biomass, and then even the soils are going to be disturbed to some extent depending on the operations. So what this means is that we can't necessarily, or we might not be able to use these other disturbances to predict what's going to happen to the forest following mountain pine beetle. So when we're trying to figure out the trajectory of this forest after it's been killed by mountain pine beetle, we're not really sure what's going to happen. So just some examples. So is this forest, after the beetle's gone through, is it going to turn into another lodgepole pine-dominated forest? Is it going to turn into something that's dominated by spruce? Or, in the absence of regeneration of any of those tree species, is it going to turn into something else that looks entirely different, that we're just not really sure of? So what we do know, so from the work of Francois Test and Vic and Simon in our department, that after the beetle goes through, there actually does develop a decent, viable seed bank, right? So the trees, they have their cones, the cones are in the air, trees die, the cones finally drop, the seeds are released, and there is actually a seed bank for lodgepole pine anyways. So then the question becomes, so given that there is seed around, are there conditions favorable for a seedling establishment? And so as most ecologists know, you have these sort of common factors affecting seedling establishment. Soil nutrients, soil moisture, light levels, the presence, the composition of understory plants, all these things are going to affect how that seedling grows, whether it survives, how it does in the future. And all these things are impacted by tree mortality following mountain pine beetle. So this is sort of a schematic I'll be using for the rest of the talk. And so this is what I showed you already. So you have your sort of attack stage is moving through through time, not town. And with that comes many different changes. So for example, the most obvious one, right? So you have needles fall off, the twigs and branches are coming off the tree, so you're going to get an increase in canopy openness, which is probably going to increase the amount of light on your forest floor. You're also going to get probably some sort of pulse and litter fall. So from a green tree moving to a red tree, it's going to lose all its needles on the forest floor, so you're going to see this sort of increase in litter deposition on the forest floor. With that, you're likely to see changes in soil moisture. So you can imagine these trees, they die, so they're not transpiring anymore, so they're not taking up water from the soil anymore. Once they lose their needles, they're not intercepting as much precipitation either. And so you might see an increase in soil moisture that probably is short-lived, right? There's that intact understory, that residual vegetation that will probably ultimately pick up that soil moisture that's freed up from the canopy. Similar to that, soil nutrients. So you have an addition of soil nutrients with that pulse of litter going into the soil, but also these trees are dying, right? So the roots are dying, they're decomposing, so that might be adding to nutrient pools as well. So you have all these changes in site conditions going on following mountain pine beetle attack. And I would argue, so those are all really, really important factors governing seedling establishment, their growth, but what needs to be added to that list is soil fungi. And so the reason for this is many. There's a lot of examples out there that show that soil fungi are important in governing tree distribution and abundance. So for example, after a series of censuses on black cherry seedlings, so Packer and Clay kind of earlier on in this decade, they showed this trend. So you don't really have to focus on these graphs. I've just sort of adapted it and summarized it in this graph here. And so basically what they found was black cherry seedlings growing further away from a parent tree. They had higher survival than black cherry trees growing close to the tree. Those seedlings growing close to the tree, they tended to accumulate fungal pathogens from the soil at a higher rate than these seedlings growing farther away from the parent tree. And of course, this had an effect on mortality. And so what this means, or what they concluded, was that sort of the presence of these soil fungi, it determined the distribution of these black cherry seedlings. So in another example, this has to do with the successful invasion of pines. So worldwide, pines have been invading various regions, say in Australia, South America. And what people are finding is that successful invasion can be attributed to the presence of ectomycorrhizal fungi. So if you bring a pine, and then actually on this topic, so lodgepole pine is one of these species that is becoming invasive in other areas of the world. If you bring that pine species into a new area, and if those ectomycorrhizal propagules are present in the soil, then that pine is likely to be able to invade. And it's invading, pretty weird places like this. So there's grasslands in New Zealand. And so with those ectomycorrhizal propagules, it can invade that kind of grassland. What also is really interesting about pine is there is a lot of literature showing that it's really, really sensitive to ectomycorrhizal fungi. So it's sensitive to the presence of those ectomycorrhizas, so are they there? But it's also sometimes very sensitive to the actual species of that fungi too. So it matters, the presence and the type of species there. Those things are both important for how pines grow. Okay, so I'm just going to tell you a little bit more about ectomycorrhizas. So this has been something I've been working on for a while, but basically, so ectomycorrhizas, they are an association between roots of most coniferous trees in the boreal forest and ectomycorrhizal fungi. Ectomycorrhizal fungi, they acquire soil-derived resources like nitrogen and water in exchange for photosynthetes produced by the tree host. So the ectomycorrhizal fungi are solely dependent on their tree host for carbon, so that's their energy source, right? And the trees, the hosts, are really dependent on these ectomycorrhizas for their resource uptake, so things like water, nitrogen, phosphorus. And so if you can imagine, these fungi, they grow around these little root tips, so they cut that little root tip off from the soil. And so there's a lot of evidence showing that ectomycorrhizas, basically, they govern the water and nutrient uptake of trees. So these associations are basically important to both of the partners. There's lots and lots of different kinds of ectomycorrhiza species. So worldwide, there's over 5,000 species of ectomycorrhiza fungi. And if you look on an individual tree or seedling, they're going to host a whole community of ectomycorrhiza fungi. So lots and lots of different species on a single host. And then as well, I'm sure you guys have all, seen this kind of picture. So this is an oak that has been inoculated with ectomycorrhiza fungi. And here's an oak seedling that hasn't been inoculated by ectomycorrhiza fungi. And this is very common. So when ectomycorrhizas are present, you often see this increase in growth. When they're not, you see a growth depression. But I want to point out that this actually is not standard. So this is the reason that we consider ectomycorrhizas to be mutualisms, but it's not always the case. And so that growth outcome, or the outcome of the association, it depends on the combination of the host species there, and it depends on which ectomycorrhiza species is involved in that association, whether, what that outcome is going to be. It also is dependent on the abiotic conditions. So, for example, in high nutrient conditions, we wouldn't necessarily expect to see this growth outcome. So I just want to make sure that's clear that you don't always see a positive growth response. Another really neat thing that ectomycorrhizas do is they form this thing called a common mycorrhizal network. And so this is a picture here, 2 seedlings. They've both been inoculated by ectomycorrhizal fungi. And you can see the fungi, it's growing this web through the soil. The fungal hyphae meet, and when they meet, they functionally connect those 2 seedlings together. And so that common mycorrhizal network has been shown to be really important for resource transfer between mature trees and seedlings. These little buttons. Okay, this picture here on the right, this is a depiction of a Douglas fir stand in BC. Douglas fir trees are these little green stars. And what this is showing you is how these trees are all connected together through this mycorrhizal network. So the pink and the blue lines, these are all mycorrhizae connecting these trees. This arrow down here, so this is pointing to a really big old Douglas fir, and it's connected to 47 other trees through this mycorrhizal network. So it's connected to big trees, it's connected to small trees, but essentially there's those functional connections between those individuals through this mycorrhizal network. Okay, so going back to our schematic, so here's what's going on in the forest. The beetle comes through, right? You have this different stages of attack. And there's going to be direct and indirect effects from this attack on the mycorrhizal fungi. So the direct effects, when you think about this tree, so it's died, when it's living, it's photosynthesizing, it's pushing or it's directing some of its carbon to supporting its symbionts, so the ectomycorrhizal fungi. But then obviously when it dies, it stops photosynthesizing, so that carbon flow is stopped. So when you think about an ectomycorrhizal fungi, this is going to have a direct impact on its abundance. And when you think about these trees, so if you remember that figure, right, they're basically hubs in this network. And if you're pulling out the hubs in the network, you might expect that mycorrhizal network to collapse or to degrade. Something's going to happen to it if you're pulling out all these tree hubs. As well, there's going to be indirect effects. So for example, going back, so you have the tree dying, it's dropping all this needles into the soil. That might change the soil conditions, and in turn, that might change the composition of the fungal communities with those changes in soil conditions. The reason why this is all important, so for someone like me who's just interested in ectomycorrhizas, I think that's super cool, but there's also other things that are really important when you think about that model, and this is about seedling establishment. So if you have these changes to the composition of fungal communities, changes to the common mycorrhizal network, and as I told you before, changes to mycorrhizas impact seedling growth. And so this is our connection here to the seedling establishment. If we see changes in these factors, do we see a change, or how does it modify seedling establishment? And that's really sort of the objective of the research we've been working on for the past couple of years. So just to sort of summarize, so you have your dead forest, and what we're doing is we're looking at a series of these ecological factors that we know are likely to affect seedling establishment. So they're going to have direct effects on seedling establishment. But then we're also taking into account that these might indirectly modify ectomycorrhizal fungi, which then alters seedling establishment too. So sort of direct and indirect effects on seedling establishment mediated through these below-ground links. And then again, where we're going, so there's variation in seedling establishment survival. This again is likely to push us on these different trajectories, what we're trying to understand. What is this forest going to look like in 50, 80 years? Okay, so obviously a lot of ideas, a lot of questions, lots of things to consider. I would just like to point out, so I've been really, really lucky. I've been working with a really amazing research team on this project for the past couple of years. First of all, so J.C. Cahill from the Department of Biological Sciences. J.C. helped me attain funding for this project, and he's really been great at helping me develop some of the ideas that I'm going to present today. Nadir, who just left, Nadir from our department here, he's been basically the brains behind thinking about the chemistry between the interaction of pines, mountain pine beetle, what might be happening to the soils, and how that would affect ectomycorrhizal communities. Suzanne Simard, she's over here on the left, right, from UBC. And so Suzanne, she's really at the forefront at leading research, understanding the ecological consequences, the relevance, the common mycorrhizal networks in forests. Janice Cook here at BioSci, as most of you probably know her, she's really at the forefront at using sophisticated molecular tools to understand that interaction between mountain pine beetle, the fungi that they carry, and lodgepole pine. We have a couple of graduate students on the project, so Greg Peck and Paul Segan, two undergraduates who are doing their own independent research projects underneath the umbrella of all of this, so Morgan Randall and Alex Sawicki. And Foothills Research Institute, so they've been a collaborator with us on this research project. And we have a series of forestry companies, the provincial government, and a biotech company that have also been supporting this research. So I just wanted to acknowledge all of them, all their help, and all their assistance on the project. Okay, so now I'm going to move on to sort of the more interesting, what have we been doing out there? So in 2011, we searched for field sites that were dominated by lodgepole pine and that were greater than 70 years old. And of course, showed some evidence of beetle activity, and hopefully we're falling on this sort of gradient that we're looking for. What we found are 11 sites southwest of Grand Prairie, and so you can see this is our red tree survey from that year. And at the time, they were kind of just on the edge of beetle activity in 2011. Since then, the beetles have come through most of our sites. These sites that we work on are adjacent to the provincial permanent sampling plots. And so the permanent sampling plots are like 4 plots, and then around them is a 100 to 200 meter buffer zone. And so with the permission of the province, we've been working in that buffer zone, so that's where we're set up. We're in the lower foothills region, and so this area is relatively wet and productive. Just to give you a sense of sort of what some of our sites look like, so just ignore Greg. So Greg was having a bad day. This was when we were out in the field, and he was collecting pieces of his field experiment that a bear had kind of devoured. But anyways, what I want you to focus on, though, so behind Greg, though, you can see this rich, abundant understory, right? So there's lots of alders present. There's lots of devil's club. Shrubs, tall, basically understory, but there was variation in our understory. So other sites, though, they were dominated more by like vaccinium species. So, so the point is not exactly the same in terms of the understory, but in the same region. The sites are dominated or in that area by Orsic gray luvisols, just to give you a sense of sort of what the soils look like. In 2011, so then we surveyed our stands to, figure out, so where do they fall in this attack intensity. What this is, so here are our 11 sites down here, and on the y-axis is total lodgepole pine basal area. And so what I want you to see, so the white denotes basal area that showed no sign of attack. The green was basal area that was attacked that year, so green attack, so 2011. The red striped is basal area that was attacked sort of 1 to 2 years from 2011. And then the gray was this more advanced decay stage, so attack that happened probably 2-3 years before we arrived. What I want you to see, so if you look at the sort of top of the colors, is that we do have this like nice gradient in attack intensity. So some of our stands are heavily, heavily intact, and then we have a series of control stands that have stayed mostly beetle-free. And that most of our stands, so that attack has happened sort of within the first, 1 or 2 years since we arrived. So pretty fresh attack. So the first thing that we wanted to find out was basically, so is there some difference in seedling establishment across this attack gradient? And so for this and for the rest of our experiments, we're working with 2 species. So lodgepole pine, so that has sort of ecological and economic importance, and then white spruce. And the reason we chose white spruce is because this is a species that we're seeing coming up in the understory. So it's already there sort of as a sapling in the understory. And so we were interested in seeing, how does it also respond to this attack intensity? So those 2 species. So what we found, so when you sowed a bunch of seeds, so each column of dots, that represents one site. What we found is, so when you sow a bunch of seeds of lodgepole pine, across this a gradient of attack intensity, you do find a decrease in establishment. And this is different than white spruce. So same thing, so if you sow a bunch of seeds of white spruce, they germinate, you follow them for that growing season, they don't really seem to be very sensitive to that attack gradient. And so this is when we thought, okay, so what's going on? So what we did is we've taken a very sort of comprehensive set of field measurements across all of our 11 stands. I'm not going to tell you about all the measurements today, or I'm not going to present results from all these measurements today. So if you're interested after the talk, if you want to come grab me and say, how did you do this? What have you found? Be happy to do that. I'm just briefly going to go over the measurements that we did take, and I'm going to highlight a few responses that we found that I think are pretty interesting. So we've taken above ground measurements and below ground measurements, right? At the beginning of the talk, I said I'm interested in these linkages. So of course our measurements span both components of the ecosystem. So we've taken measurements for needle deposition. So what we do there is we throw out litter traps at each of our stands. We go and visit them at multiple times during the growing season, collect the litter, weigh it, or dry it and weigh it. We're also looking at the quality of that litter, so not just how much has come down, but also the quality in terms of the nitrogen and phosphorus content of those needles. And then we're also looking at decomposition rates of those needles across our stands too. For light, we've measured this in 2 different ways, so in terms of canopy openness, and then we also have a light meter that we can take those measurements on the ground. The understory, we're looking at, so the species composition of the understory, including shrubs, and also looking at the biomass of the understory, so sort of getting a sense of how abundant it is. For the below ground measurements, we're looking at soil moisture. So again, we come out multiple times during the season, measure soil moisture across all our stands. Soil chemistry, which includes sort of a bunch of things. Soil nutrients, we use those plant simulator root membranes, so those PRS probes, to measure soil nutrients across our sites. We're also looking at soil phenolics. So you guys might be like, weird, soil phenolics. So Nadir and Paul are really leading that aspect of the study, and I'm just going to say a sentence about it. I won't really be presenting anything on it today. But the idea behind the soil phenolics is, so you have these trees, right? They have needles, and in those needles contain a lot of secondary metabolites in terms of, so the tree can defend itself. Those secondary metabolites, one of them, or one compound is these phenolics, and they don't degrade very easily. So when those needles, so when the tree dies, the needles fall to the forest floor, those phenolics are still contained in those needles. And so you can imagine that if you get a whole bunch of needles in the soil, that might change the abundance or the concentration of phenolics in the soil, and that might have an impact on soil biota like ectomycorrhizal fungi. So that's sort of the reasoning behind the soil phenolics. Also measuring pH, and then moving to sort of the biotic parts of the below ground ecosystem, we're measuring roots. So of course you have all these dying trees, so you expect a change in the abundance and density of roots in the soil. And then finally, my favorite, ectomycorrhizas. And we're measuring ectomycorrhizas in a variety of different ways. So we're measuring hyphal abundances in soils. We're looking at colonization rates in roots. We're looking at sporocarps, which I'll talk about in a few minutes. And we're also using next generation sequencing technology to characterize the ectomycorrhizal communities that we find in soils. And so this is using the ion torrent that the MBSU purchased recently, or I guess about a year or two ago. Okay, so that's everything we measured. Again, I'm not going to present all of that, but here are some interesting things so far. So one of our core ideas in our sort of working model is, of course, that these trees die and they drop a bunch of needles, and that might matter. And we're finding data that does support this. So on my graphs, I have mountain pine beetle-killed basal area. So basically this is our attack intensity. This will be on the graphs following. On the Y is litter mass. So this is needles that we collected in our traps. And you see this positive relationship, right? So it's really obvious. Attack stands, there's more needles coming down than in control stands. But this effect appears to be transient. So this is what I just presented here was 2011. Here's what we found in 2012. So that trend disappears. And so what we think is going on is, again, there's just this short window of time where you get this large input of needles. And I should mention that we find evidence that the needles coming down from attack stands, they tend to be more nitrogen rich than needles coming from the control stands. So you have this short window of time, a lot of needles are coming down, have more nitrogen, so this is likely to affect the soil conditions. Soil moisture, this tells sort of a similar story. So same thing, attack stands have about 10%, or I guess are 10% wetter than control stands. But this effect is transient. So this is what happened in 2011. We went out and resurveyed in 2012, and there's no trend. So again, it's this very short-lived sort of pulse of resources. And what we think is going on is as these trees die, so they're not transpiring, they're not taking up this water, the needles fall off, they're not intercepting so much precipitation, but what happens is that residual vegetation is picking up that water. So it's available one year, it's gone the next, and this data would support that idea. So this is a measure of herbaceous biomass. So we went, had square meter plots, cut all the vegetation in that plot, dried it, weighed it, and that gives us a measure of biomass, that plant biomass. And you'll see that there's this increase. So attack sites have more biomass than these the control sites. Okay. So I've talked a lot about ectomycorrhizas, so I want to tell you a little bit about what's going on with the ectomycorrhizal part of this system. So Roland True, he joined our research group last year. Roland True is a mycologist from Athabasca University. And he went out and he sampled all our stands. He basically set up transects and identified the sporocarps that he saw along those transects. And so sporocarps, they're the fruiting body or the fruiting structure of a type of fungus. And so ectomycorrhizal fungi, they'll form sporocarps. Decomposing fungi, they also form sporocarps. And what he found is that across the 11 stands, on average, there's about 30 species of sporocarps. And this doesn't change with attack intensity. And so the more interesting part of that story, though, is this. So if you look at the proportion of those spore carps that are ectomycorrhizal, you'll see that that goes down with attack. So our control stands, they had about 60% of those spore carps are ectomycorrhizal. The attack stands, that drops to about 10%. So we're seeing this substantial decrease in the reproductive output of these ectomycorrhizal fungi. We're following up on this work. So Morgan Randall, she is assessing hyphal abundance in soils to see if that matches up with this trend as well. She's looking at percent colonization of root tips, again, to see if it's telling the same story. And then we'll be characterizing the ectomycorrhizal fungi across our soils in our stands to see if there's also a shift below ground in the species that we find. So hopefully more next year on this point. Okay. So moving from the field and into the greenhouse. So the field, obviously, there's a lot of simultaneously changing conditions. And so what we want to do is try to isolate the effects of the ectomycorrhizal fungi from all these other things that are changing. And so the way that we've tried to do that is in this experiment where we're isolating the effects of the fungi from litter and light. And so I'll go through each of these. So what we've done is we've set up a factorial experiment where we have a bunch of pots and we grow either lodgepole pine or white spruce in these pots. The pots contain a common sterilized substrate. To those pots, we add either no pine litter, litter collected from our control stands, so these are the stands that haven't seen any beetle activity, or litter from our highly attacked stands. Crossed with that, we add soil inoculum either from our highly attacked stands, our control stands, or no soil inoculum. And then to that, so as I mentioned, we're using lodgepole pine and white spruce. So lodgepole pine is an intolerant light species. White spruce is more tolerant to shade. So we've crossed this with two light levels corresponding to what I'm going to call full light and shade. The full light loosely corresponds to light conditions over the understory in our attack stands. The shade sort of loosely corresponds to light conditions above the understory in our control stands. So that's kind of what we were gauging for. This is just sort of what the experiment looked like. We had, so we let these seedlings grow for 8 months. Half of the experiment we harvested, and we are measuring those seedlings now, so we're weighing them, so we're assessing their growth. We're also looking at which fungi colonizing the roots of those seedlings. The second half of seedlings we put out on the roof on the greenhouse, and so they experienced sort of a natural dormancy. We left them out in the fall for a couple of months. Those seedlings that experienced that natural dormancy, this is what I'm going to present on. And so for those seedlings, we have total biomass right now. And then we are doing some other measurements, but I don't have those in there today. Okay, so for white spruce, here's a total biomass of spruce grown under full light and shade. I'll tell you right now, so litter did not make a difference. We're 99% sure litter it didn't matter. So I pooled that data, and what you're looking at, so this is soil inoculum from our control stands, from our attack stands, and seedlings grown with no inoculum. So right away you'll see that, so white spruce seedlings grown without any inoculum, that causes a sort of a growth depression, right? So we kind of know this already, that if these seedlings don't have any ectomycorrhizas, they're not going to grow very well. If you give them some kind of inoculum, they do a little bit better. If that inoculum comes from control sites, they seem to do the best, but of course there is some variation around that. For lodgepole pine, it's a similar story. So the graphs or the bars are set up in the same way. First thing I want you to notice, so when lodgepole pine seedlings are grown without any inoculum, they really, really suffer. So this agrees with what I said earlier on in the talk, right? Pine are really, really sensitive to the presence of ectomycorrhizal fungi, and our data support that. If you give those seedlings some kind of inoculum, regardless what it is, they do better, right? These bars are higher. And it looks like under some conditions that the lodgepole pine actually is really sensitive to the source of that inoculum. So it matters if it comes from the control stand or the attack stand. It has implications on seedling growth in this situation. This is all pending final analysis. I don't want to get super excited about it. But it does look like there is something going on, that the soil biota, those ectomycorrhizae fungi, do matter in terms of seedling growth. Okay, so one thing in a greenhouse experiment that you can't do is you can't really account for the effect of large trees on seedlings through that common mycorrhizal network, right? We talked about that at the beginning of the talk. So this requires to go out in the field and set up an experiment. Here's a picture that I showed earlier. So here's 2 seedlings inoculated by ectomycorrhizae fungi, right, forming this network. And what you can do is you can manipulate the degree to which that seedling connects into that network through the use of these mesh bags. These are nylon mesh bags. Those nylon mesh bags, they differ in their pore size in the fabric. And so what we've done is using those mesh bags, so sorry, let me back up. You take this mesh bag, you fill it with a bunch of soil, and you put a seedling inside it. And then that is going to manipulate that degree of connectedness to the mycorrhizal network. We've set up 3 treatments. One that excludes mycorrhizal fungi and roots, so nothing can grow, nothing can penetrate those bags. The second treatment allows for fungi to grow through those bags, but no roots. And then the third treatment is basically a handling control, where we dig up the soil and then drop it back down, and then obviously roots and fungi can grow through there. So 3 treatments we've set up for both our species, lodgepole pine and white spruce. These treatments are replicated 10 times at each of our sites, so 11 sites. And just to give you a sense of kind of what those look like. So here's a picture. So we dig the soil, we put it into the bag, we try to keep those soil horizons intact in the bag, try to get the soil sort of as flush as we can. Here's a picture of 2 of those bags, so installed in the soil. Then we put some seeds of either lodgepole pine or spruce into those bags, let them germinate, and then sort of it looks like that later on in the season. Should also mention, so what we're doing is we use our soil moisture probe and we take measures inside and outside of the bag to account for any, biases towards soil moisture. We haven't found any so far. And what we're doing with that is basically, so I don't have results from that to present today. So those seedlings we're going to harvest at the end of this year. And so we should find out sort of what's going on with that. But what we're looking for, so I've just sort of depicted what we're imagining. So on the y-axis is seedling growth differences. So when a seedling is connected versus not connected to that network. So looking at the differences in growth between those 2 situations. Here's our attack intensity. If there's minimal to really, really low differences in growth, then we would say, so that common mycorrhizal network is not really important, and it's important doesn't vary across its attack intensity. If we have the situation that those growth differences between those 2 situations are really, really high, then we would say that that common mycorrhizal network is really important. If we have this sort of situation, that it changes with attack intensity. So this is telling us that that importance is changing through attack, or it could be saying that that common mycorrhizal network is actually degrading across this attack intensity. And we'll be using our molecular tools to ID which species of fungi are on those seedlings so that we can sort of discern between those 2 possibilities. So again, this data hopefully will be next year, but it's coming. Okay. As I was putting together this talk, being all nervous and agonizing over the slides like I will, I wanted to have a really, really nice, tidy slide at the end that said, we know everything, here are all the answers. But I couldn't put it together, so not surprising. So instead, what I have told you is, so I introduced some of these core ideas that we have about what is going on with this system. After the pine beetle goes through, the trees die, we see all these changes. I presented evidence to support some of these core ideas, right? You get this pulse in litter fall, you get this pulse in soil moisture. We're finding that, and it happens very quickly, but it's transient. I didn't present data on this, the gap extent in soil nutrients, but I can tell you that we are finding data that also supports this model. I'd like to think that we're building evidence to suggest that ectomycorrhizae fungi are important in impacting seedling growth in this situation. So they are a piece of this puzzle. But in fact, I don't want to leave with this schematic, right? This is really simple. We have these different factors or responses, depending on how you think about them, acting independently, sort of one at a time on seedling establishment, no linkages in between, right? And instead, I think what our data is showing as we sort of move through this project, that it looks something more like this. So you have this single event. You have a bunch of beetles attacking a bunch of trees, and these trees die. And then what happens is that you get this cascading web of responses, is more how I think about it. And so you have indirect, you have direct responses, but they don't necessarily all act in isolation, right? So some of these lines, they're mediated through other factors. There's above and below ground linkages. At the end of the day, we might find that, in fact, some of these factors aren't so important, but this is sort of where we're going with the project right now, is sort of putting it in this framework. You have this cascading web of responses and trying to tease out which ones are important, which ones do we need to think about, which ones are going to be important for management. Where we're also going with this project is to be thinking about the temporal dynamics. So in my presentation, I showed you 2 data points pretty much, right, in 2011 and 2012. And I'm calling some things persistent effects, some things might be transient, but with 2 data points, that's really hard to say. So we have another survey coming up this year, and what we're pushing for is to keep monitoring this site for the next few years in the future so that we can account for that variation in time and with our different measures. Also, what I think is really important is to incorporate spatial dynamics. So on the landscape, you have some areas where just a few trees are attacked. You have some areas where patches of trees are attacked, and other areas where you do have these full stands that are dying. And I think it's really important to sort of acknowledge that, and then maybe to be looking for, say, thresholds. So is there some spatial scale threshold that we need to be thinking about soil biota? Or is there some scale that we don't have to worry about it? And these are sorts of things that we're going with this project in the future. So and then again, just to remind you why it's important to be looking at seedling establishment and trying to figure out what's controlling seedling establishment is to predict these forest trajectories. Where are our forests going to go? What are they going to look like in the future? And on a side note, what do we want them to look like in the future after mountain pine beetle has gone through? So with that, I just want to say thanks to a few people. So the graduate students, Paul and Greg, tremendous amount of work on this project, and they've been really, really great about handling all this. Morgan and Alex have just been inspirations as undergraduate students, coming up with really, really great questions, and again, working so hard on their projects. Roland, he came in, as I mentioned, exceptional mycologist. We had a series of helpers in the field, Mike Devine, Brittany, Patrick. Mohammed has been helping us in the lab, and we have new BFFs with Sophie and Corey Davis in the MBSU. They've been helping us tons with our molecular work. And then finally, Grand Prairie Regional College. When we're up in Grand Prairie, they've been really gracious and be letting us use their facilities for a lot of our work. So with that, I guess I'll take any questions. Thanks.